Welcome to this video on the anatomy of cells. We will explore eukaryotic cellular structure, function, and the mechanics of homeostasis. Homeostasis is the maintenance of a stable internal environment, is a basic biological necessity. Changes in cellular processes can lead to disease and aging. What you see here are the basic steps, which you should be familiar with for the next exam. During the semester, we will be going through specific examples of homeostatic mechanisms, such as blood clotting, blood pH, and bone remodeling, and more. The cell is the fundamental unit of biological logistics. Key tenets of cell theory include that all living organisms are made of cells, cells are the smallest units of life, and all existing cells are derived from other cells. All cells also maintain homeostasis. Understanding the basics of cells will be important as we look at modified cells this semester, such as muscle cells, bone cells, and neurons. Organisms are classified as either prokaryotes or eukaryotes. Prokaryotes are generally smaller, lack a nucleus or organelles, and are always single-celled. Eukaryotes are larger, have a nucleus and organelles, and can be single or multicellular. Since we are studying humans in this class, we will focus on the eukaryotic cell. Keep in mind that when you take microbiology, you will be learning about prokaryotic cells. Cell size is limited by surface area, which is necessary for nutrient absorption and waste removal. As cell volume increases, the surface area to volume ratio decreases, making survival difficult. The plasma membrane is a fluid, selective barrier. The bilayer, made of phospholipids, provides physical separation and structural connection. The hydrophilic heads face outward, and the hydrophobic tails face inward. Embedded proteins in the membrane act as communication and transit hubs. They function as receptors for signals, enzymes to increase reaction rates, carriers for transport, anchoring points, and recognition markers. The cell membrane is selectively permeable. Small nonpolar molecules can pass freely, while large polar molecules need protein assistance. Passive diffusion moves substances down their concentration gradient, from high to low concentration. Osmosis is the movement of water across a semi-permeable membrane, driven by differences in solute concentration. Water moves through aquaporins from areas of low solute to high solute concentration. Water is always moving in and out of cells through these pores. Keep in mind that this movement is based on two concentrations: the concentration of the fluid inside of the cell, intracellular, and the outside of the cell, extracellular. All cells have fluid inside and outside/around them. Let's look at what happens when there is a difference in concentrations around a cell. In a hypertonic environment, the cell shrivels. A hypertonic solution means that it is more concentration, more solute, and less water. An analogy would be like really strong coffee. Coffee molecules are solutes in the water. Hypertonic solutions around a cell will pull water out of the cell to equalize the concentration. In an isotonic environment, the solution around the cell is the same as the internal solution. In a hypertonic environment, the solution around the cell contains more water and less solute. Think weak coffee in this case. So the water is pulled into the cell, which can cause it to burst. Understanding these concepts is important for nurses, as this is the basis for the types of solutions you choose when you give someone IV fluids. Transmembrane protein channels enable two types of transport: passive and active. Facilitated diffusion is passive, moving with the gradient without energy. Active transport moves against the gradient and requires ATP energy. Essentially, molecules move from high to low concentrations when using diffusion or facilitated transport. You will see this through your A&P classes. It is especially evident when you get to A&P2 when learning how oxygen moves from the lungs into the blood. Vesicular transport moves bulk cargo by reshaping the membrane. Exocytosis moves material out. Endocytosis moves material in. Phagocytosis is a specific type of endocytosis where cells engulf large particles. Phagocytosis is how some of your white blood cells engulf foreign cells or clean up cellular debris. The cytoskeleton provides structural integrity and motility. Structures like microvilli, centrioles, cilia, and flagella are all components of the cytoskeleton, supporting different cellular functions. We will see this come into play this week when we review mitosis and meiosis, the two types of cell division. Organelles are specialized nodes within the cellular factory. They are grouped by function: protein production and packaging, synthesis and detoxification, energy generation via mitochondria, and information control within the nucleus. We will look at a case study in a separate video to go through the organelles and apply them to disease. Anatomical structures aid physiological processes to maintain homeostasis. Different cellular components function as an engine, filters, and logistics to ensure the cell's internal balance is maintained.